Thank you, Jason. All right, so what I'm going to talk to you about for the next 20 to 25 minutes is actually a slightly different topic, which is gene expression and how we can relate that to brain function in terms of looking at network behavior. So just to put this in context, we've learned a little bit about the history of imaging genetics, the modern day endophenotype, how to use big data sets, and then Sylvain recently, or just before the break, talked about imaging epigenetics. So I'm going to talk about, again, something new to this course at least, and to the field in general, which is trying to relate gene expression in the brain to brain networks, especially functional networks. So I'll give you a brief overview of gene expression, then talk about how we can relate gene expression data to functional imaging networks, and then talk about essentially 3 papers that have come out recently that have attempted different ways to do this, and then close with summary and interpretations. And the main take-home message here is that this is a new field. So those of you who are graduate students or postdocs or even PIs and are interested in pursuing something relating gene expression to imaging phenotypes, this is a field that's sort of just exploding, partly because of the availability of new technologies, but also some of the availabilities in the experimental paradigms and availability also of tissue samples. So this is an expression of, an overview of gene expression. So how many of you are familiar with gene expression data? How many have actually worked with this type of data? Okay, some of you haven't. For some of you, this might be completely new. You might not even know what this means. So I've put together a little slide to talk about what we mean when we talk about gene expression data. So gene expression data is essentially the quantification of messenger RNA in a biological sample. So we're looking at a specific class of molecules, gene products, mRNA, that you can find in a specific sample. So this could be a tissue sample. More recently, there's been an explosion of trying to identify and quantify mRNA in single cells. So this is a field that's rapidly advancing and generating a large amount of data, and even the analysis techniques for this type of data are still in flux. So let's take this as an analogy for those of you, again, who might still not be fully comprehensively aware of gene expression data. So in every cell we have a genome, and you can think of this as sort of a dictionary or a dictionary plus grammar. So it contains a set of all the words, all the molecules that a biological system can actually generate, and in addition contains rules about the interaction of some of these molecules with each other in terms of binding sites and also in terms of epigenetics, as Sylvain has just described. Genes can be considered the analog of individual words, so they're all contained in the genome, but the gene expression profile is a specific story that comprises or is made up of these words. So the idea is that different cells or different tissues in the brain and in the body and different organisms will have different proportions of these words or gene expression products in them. So the idea behind gene expression profiling is you actually look and you count up or you assess the relative quantity of these words or gene expression products, mRNA, and then you try and infer relationships between them, or you try and tie that to the function or the composition of a specific tissue or type of cell. So just for fun, this is the type of data that you would get from gene expression, except this is not gene expression, this is literature. So this is a word cloud from a specific book or story, and the size of the word indicates the frequency of that word in the story. So the idea behind gene expression analysis is you get this type of data, so relative quantification of various gene products, and the idea is to infer something about the sample that you can actually get. So in this case, does anyone know, or can anyone guess what story this word cloud could be from based on the frequency of its words? I know we're scientists, but let's get some literature in here. Yes, please shout it out. Yes, this is from Les Misérables by Victor Hugo. And you can tell that because there are words that are specific to the story. If you're familiar with the story, you'll recognize Marius, Cosette, Valjean, so all of these characters in this book. Here's another one. Any guesses? I think I heard it. This is The Jungle Book by Rudyard Kipling. And one last one that's a little more difficult. Okay, maybe not that difficult. This one's Beowulf. So the idea is that from the gene expression, what you essentially get is the frequency of these molecules or words, but you don't actually get direct information about the relationships between these words. You can't reconstruct a sentence based on this. And you also don't have any information about the underlying motives or themes that the authors of these 3 books were trying to create. That's where the picture gets more complicated and you have to look at interactions among genes or epigenetics. But the basic idea is you take a tissue sample or a single cell and you identify all the products or mRNA that's in it and their relative quantities. So the idea is you can actually take different samples, and this is the important part. So whereas in the epigenetics talk that Sylvain just described, one of the issues is that there's heterogeneity in the epigenetic profile of different tissue samples. For gene expression, we actually take that bug, so to speak, and turn it into a feature. So one of the things we want is actually heterogeneous gene expression profiles from different tissues or different cell types. And this variation then allows us to try and infer relationships among genes, either based on correlation or more complicated statistical measures. So gene expression data, for those of you who haven't seen what that looks like, you get for each gene, in this case each row, a numerical value for each of the samples or the regions that you've been profiling. So the idea is you get this giant matrix that's just a set of numbers describing the relative amount of each gene in each sample that you have. So it's a fairly straightforward data modality that comes out from gene expression, and there's multiple techniques to profile them, but they all give you the same essential ultimate data format. And the challenge now is to relate the gene expression data on the left here to some sort of image modality. So most often what you'd have to do is, because these genes come from specific regions of the brain that have been dissected post-mortem, you can actually map them to an image-based parcellation of the brain. So you use imaging-based coordinates. The most common one is MNI coordinates. Obviously, for post-mortem samples, for gene expression, you don't get functional MRI or functional data. You only get structural data. But that's often sufficient to be able to register these gene expression samples into some imaging coordinate framework. The next question in relating imaging and gene expression data is you have to figure out what metric you're going to use to correlate or relate the quantified gene expression data to the imaging-derived metric. So you have to choose something on both sides, from the gene expression side as well as from the image-based the imaging data side. So the papers that I'm going to talk about have basically can cover 2 different strategies. So one is on the left, on the lower left, as you can see, the gene expression data for a single gene will vary over the different regions that you've sampled. And so if you want to do a direct correlation, you need an imaging metric that also varies among the regions that you've sampled. So you can define an imaging metric. We'll look at one specific example of this. And then you can do a direct relationship. Correlation is probably the simplest measure to try and identify on a gene-by-gene basis which genes seem to have significant correlation with your imaging-based metric. However, oftentimes with functional imaging data, what you get is not some measurement for each specific region, but rather a relationship among different regions. So if you're talking about functional connectivity, what you get is a correlation between either voxels or parcellations of your image space, or your imaging space. And the idea then is somehow to relate gene expression to the overall functional connectivity that you've obtained from your imaging data. So this is a little more challenging, and we'll talk about a couple of methods that can be used for this as well. So these are the 3 papers that all came out recently that I'm going to give you a quick survey of. Again, you can look at these in more detail. These slides will be available, so you can find these papers and read them in more detail. But 2 of them that I'm going to talk about use the same gene expression data set. So this is from the Allen Human Brain Atlas, and it consists of 6 brains with about 500 postmortem tissue samples per brain, of which about half are from the cortex, the other half are from subcortical regions. And so both of these studies that I'm going to talk about use this gene expression data set. In one, they have an internally generated functional imaging data set, just fMRI, looking at 4 specific networks. So the default mode network and then 3 others. On the right, the other paper that I'm going to talk about looks at the parcellation or the connectivity map that came out from the Human Connectome project. So all of this data is actually available for download. The Allen Brain Institute has an interface to navigate some of this data, as does the Human Connectome project. So it's easy to look through this data if you're interested. So the first step, as I pointed out, is how do you align the samples from which you obtain gene expression data to the actual imaging space? So this is just from the first paper that I'm talking about by Jonas Ricciardi and Andre Altman that came out a couple of years ago. In this, they used the MNI coordinates from the gene expression data from the Allen Institute and then registered those samples into their actual imaging brain space. And what they can then do is associate each of these samples, which is represented in a circle, with one of the networks, one of the nodes of the networks for which they've actually identified through the fMRI data. So once that's been assigned, the question is, how do you correlate the gene expression variation in this brain or the set of brains with the imaging data? So they use something called the strength fraction, which intuitively is very simple. You look specifically at a network defined by the imaging data, and you say, what is the correlation of the gene expression among the nodes within this network? And then you compare that to the correlation of the gene expression profiles of the nodes that are outside the network, again, the network having been defined based on the functional imaging data. So then you can go and look to see, first, what they find, let me say, is that there is a correlation. There is an enrichment of gene expression correlations within a network versus outside. So there's some relationship between the gene expression data and the imaging data. But because what we're doing is we're getting correlations among these regions based on all 20,000 genes. So the idea is that even if there's a correlation between two regions, or you find that, that nodes within a network seem to have more similar gene expression than nodes that are not in the network, you still have to assess individually which genes are contributing most strongly to that relationship. So they actually generate a consensus list of genes most strongly contributing to the strength fraction within each of these networks. And they do that by taking each gene, removing its information, and also upweighting its information and seeing what the change in the strength fraction is. And this is a list of the genes that they derive as part of this consensus list. Consensus means, again, since the gene expression data comes from 6 different brains, they're going to look across all 6 brains and see if the same genes keep contributing strongly to the strength fraction. So these are the list of genes that they come up with. In most gene expression analyses, one of the first things you do is you go and look to see in the gene ontology if these genes belong to a specific class or category. So in this case, they find that a lot of the genes belong to the ion channel category and then are associated with disease. Again, not so surprising, gene ontology analysis tends to be a fairly blunt instrument and tells you things that are pretty general, but it's a nice confirmation that these genes are at least have some relationship to known brain function. So they also look now to see if they can find these consensus genes in other contexts. So that one of the validation analyses that they perform in this paper is to look at the Imagen data set and then look to see for each individual in the imaging data set, in the Imagen data set, they actually identify or calculate a genetic score which is related to the variability in the genetic information among this consensus list, this top list of 136 consensus genes. So if there's an individual that has no SNPs in any of these genes, they're going to get a low genetic score. If there's an individual that has SNPs in multiple of these genes or variation in multiple of these genes, they're going to get a high genetic score. And so the idea is if you look at individuals with a high genetic score, in other words, where these consensus lists of genes have been perturbed versus those with a low genetic score, in other words, individuals in which there's very little genetic variation in the list of consensus genes, they can then look at the imaging data from each of these individuals and identify the difference in the relative strengths of the connections in each of these networks. So the idea is that if these genes had nothing to do with connectivity in general, the randomness of connectivity weight differences from individual to individual would not be related to whether or not they had changes in these specific genes. So what they find is actually the opposite, that in individuals with high genetic scores, you get dramatic increase or variation in the weights within each of these networks, whereas with individuals with low genetic score, it's essentially very little to do with these variations in the genes seem to have very little to do with the correlation of the strength of these connections. So it's, again, a fairly global analysis, but the idea is to look and see if these genes have anything to do with connectivity. Finally, they also look in the mouse, which, as we've pointed out, is perhaps not the best model system for studying human disease, but with the aspect of looking specifically at structural connectivity or functional connectivity in non-disease models. The idea is you can actually look at the relative weights of the connectivity. This is actually from structural connectivity analyses between various cortical regions. And then you can look at the transcriptional or gene expression similarity of those same regions. And just by eye, you can actually see that there's a clear link between structural connectivity versus transcriptional relationships using this list of consensus genes that they find. So the other paper, which is actually related to this, which came out from the Allen Institute last year, is to look from a different axis. So when you start with the gene expression data and then try and relate it ultimately to the functional data. So the gene expression data, the basic idea is you try and identify genes that show the same differential expression patterns across tissues over multiple individuals. So for example, this gene, protocadherin 8, you can see that in each of these 6 graphs, that's each represents a different individual, the overall gene expression pattern is actually very, very well recapitulated. This is not the case for all genes. There are genes that show high variation among individuals, but the focus of this paper was to look specifically at genes that show very, very conserved differential expression patterns over different individuals. And you can actually score every gene using a metric that we call differential stability, and then look at those genes and see specifically what types of expression patterns they have. I'm not going to go too much into this because this just shows, again, that different modes or groups of genes have very stereotyped expressions that seem to be cell type or region specific. The main point is that different regions of the brain in the same individual have dramatically different gene expression profiles. So if you look at the differential stability, the genes that are very conserved in their expression pattern across individuals, and then you actually look to see how their expression pattern looks across these different regions, what you find is on the y-axis here is the relationship between the functional connectivity and the gene expression similarity. And then on the right is the stability or how reproducible that gene's expression is over individuals. And the overall correlation scores, as you can see, are quite low, but the general trend is clearly visible that genes that are more preserved in their expression across individuals tend to be more related in terms of their regional specification to actual functional connectivity. So again, a very global look at this. The genes highlighted here in red are actually the same list of 136 consensus genes from the Ricciardi paper. So you can see that a lot of those are both stable and have high correlation with functional connectivity. So the third paper that I'm going to talk about, again, very briefly, is to relate gene expression to the default mode network. And this is a paper by Wang and Bellegarde again, 2 years ago. And the idea is here not to try and relate things to the connectivity between different regions, but to just directly compare the gene expression value to some measure or metric that can be identified across all the different brain regions in the functional imaging. So they come up with this metric called FALF, the fractional amplitude of low-frequency fluctuations. So this is the power that's contained in the low-frequency fluctuations in each region of the brain. So what you can do is then take each gene, so you have its expression profile over multiple regions, you have the FALF over multiple regions, you can actually do a direct correlation between the two, identify the genes that have the highest correlation with the functional data, and then look to see what those genes are. This is just a graph showing that the observation for the number of functional genes or number of correlated genes appears to be higher than what you'd expect by chance. These are just from shuffling the data in each of these data sets. On the left, it's a little hard to read, but this just shows that when you use 2 different fMRI data sets, one is from the 1,000 Connectomes project, the other is from the New York study, you can actually get very reproducible results based on both which gene expression data set you use, but also which fMRI data set you're using. So on the X and the Y-axis are just the 2 different fMRI data sets. You can see that genes with high correlation in one tend to have high correlation in the other and vice versa. And this is a list, again, of the genes that they found with the highest correlations. They're neuronal related. Some of them are implicated in brain-related disease. But again, that's the level of information so far that we can glean from comparing gene expression to connectivity. So the main findings at this point are that significant correlations exist between the gene expression data and functionally derived data. However, these genes tend to belong to some very broad categories, ion channel or brain-specific, sometimes associated with disease. And the variation in these genes is related to functional connectivity. But this is where it gets interesting, and this is, if you remember nothing else from this talk, I hope you remember this, is that this is a field that's still in its infancy. So these results are really, really broad, and the only thing that they've really shown, in my opinion so far, is that there's some relationship between gene expression data and connectivity-related data. These don't cover any disease phenotypes, they don't cover high-resolution imaging, but the idea is that with all of that information on the horizon, it might become possible to do much more sophisticated analyses and use gene expression as another data modality to try and identify molecular mechanisms behind variations in either brain structure or variations in brain activity due to disease or even in healthy patients. So the possible interpretations of this are, I won't dwell too long on this because Jason will talk about how we interpret some of these results. And there's three possibilities because all of this is just a correlation-based analysis. There's no causality here. It's very difficult to identify any functional relevance directly from these genes. But it's possible that the gene products are responsible for maintaining networks. It's also possible that the network mechanisms might drive the expression of these genes. And it's also possible that just because these genes are neuron-specific, that these networks are influenced by neurons, and so you find that these genes seem to have high correlation. So what have we learned? As I just mentioned, the field's wide open. The initial conclusions are very broad, and there's actually a couple more papers that you can look through that look at a little more sophisticated analysis, looking specifically at connectivity statistics and their relationship to genes, as well as looking at oscillations during memory coding, and then looking for genes that seem to be correlated with these types of functional data. But this is the most exciting part, what's on the horizon. So there's a number of things that are coming up. There's new and better parcellations of the brain based on both functional and structural imaging data. There's higher resolution measurements coming both on the gene expression side. I already mentioned single-cell gene expression is now becoming a staple of the field, but then also on the imaging side. Then one thing that you might have noticed, or if I haven't said it explicitly, I should say this now, one again, feature or bug is that you're using brain tissue and you're taking advantage of the heterogeneity of brain tissue to establish relationships with connectivity. So that's nice because the cellular or brain-specific heterogeneity is actually something that you're using to your advantage. The disadvantage is you're not getting the same imaging data most of the time from the donor that's giving the sample because one is a postmortem procedure and one requires a living individual. However, even that's changing because now there's a number of studies out there. The one that comes to mind is the ROSMAP study, which contains imaging data, clinical data, and then now as the participants in the study are donating their brains, you're actually starting to get some gene expression data. So this is on the horizon and something that will really provide this field of relating gene expression and connectivity a whole new axis because you'll be able to study disease because you'll have these matched samples. Finally, there's also other axes along which these analyses could be extended. You can look at subcortical regions. The gene expression data for that actually already exists. You can look at the relationship between gene expression and endophenotypes. Again, this requires some sort of matching between the samples that you're getting for gene expression and the imaging data or whatever endophenotypic data you're using. So just as a conclusion, again, if you take nothing else from this talk, that there is a relationship between gene expression and functional connectivity, not a big surprise, but more interestingly, this field is on the horizon. There's going to be a lot more cool analyses and things and experiments to be done in order to establish some more of the molecular mechanisms behind the establishment of functional connectivity and also the dysregulation of connectivity based on gene expression. Thank you.
Thank you so much, Vilas. As David comes up, any questions for Vilas? Yeah, do you mind going to the microphone? Thank you.
Thanks for your talk. So a lot of the studies that have been done so far have looked at the adult gene expression maps. I just wondered if you had any thoughts about ways one might be able to work with the developmental data that can kind of deal with the lower spatial sampling, I guess, the spatial resolution.
Yes, that's an excellent question. So there do exist some developmental data as well on the brain. Obviously, acquisition of that tissue comes with its own challenges, but the Allen Institute, for example, has a developing atlas of the human brain. So you have gene expression data from different brain regions through development, both fetal and then early adult. And that's actually a very important point because when you're trying to establish relationships between connectivity, especially if you're talking about structural connectivity and gene expression, a lot of that might actually be laid down in development and not present in the adult. So it's an important other additional axis to consider. One final thing I will say, because again, this is a new thing that's coming out, is there's a new field of looking at or generating sort of brains or quasi-brain tissue in the dish. And so that is an important aspect also to try and identify very early developmental stages. It's still a very young field, but the idea is that you can actually take cells from a living individual, convert them back to stem cells, and then differentiate them into brain-like or very immature brain-like structures. And so then that provides another avenue to try and access some of the developmental data that might not be available for obvious reasons, from human tissue. But yes.
Okay, thanks again, Vilas.